Today I will be presenting some recent findings from our lab. The title of the talk is Trans-Valvular Unloading Reduces Anaerobic Glycolysis Before Reperfusion and Preserves Energy Substrate Utilization Post-Reperfusion in Preclinical Model of Acute Myocardial Infarction. Okay, so, sorry. So these are some of the grants I got in past to do this research. So our research from our lab has already shown that transvalvular unloading or LV unloading reduces LV wall stress and reduces infarct size despite delay in reperfusion in preclinical model of acute myocardial infarction. And in our recent work, we found out transvalvular unloading does so by preserving myocardial energy substrate utilization and protecting mitochondrial function. For this study, the hypothesis is LV unloading reduces infarct size and promote myocardial recovery by first reducing ischemic injury and then further reducing or limiting the reperfusion injury. So before going in detail about ischemia and reperfusion, I would like to go back to the molecular mechanism, what happens during ischemia and then post-reperfusion. So during ischemia, what happens is there is a limitation of oxygen to the tissues or the cells. So to that, the myocardium response is to handle the ischemic signaling. So we wanted to understand how LV unloading is affecting this ischemic signaling. Is it any way different than normal ischemic tissue, or if it is, then how it is? What's different? And the second part is the metabolic changes. So as soon as the tissue or cells senses ischemia, they switches to anaerobic glycolysis. So increased glucose uptake, increased lactate, and which lower the pH in the cell, and that leads to closed MPTPs. And upon reperfusion, when there is a huge flush of oxygen, that leads to washout of lactate and leading to MPTP opening, which leads to increased ROS, increased calcium handling or calcium overload in the cells, leading to cell apoptosis and necrosis. So keeping that in mind, we wanted to understand the molecular signaling, oxygen signaling in the cell. So the molecular sensor in the cell is known as hypoxia-inducible factor 1 alpha. So how it senses ischemia is in the presence of ischemia, or in the presence of oxygen, PhDs, the enzymes prolyl hydroxylase domain enzymes hydroxylates HIFs, and it is degraded. But when there is absence of oxygen, when there is absence of oxygen, PhDs could not hydroxylate HIF. HIF started accumulating in the cell. That leads to, then the HIF is translocated into the nucleus where it makes a dimer with HIF1 beta, and then the HIF responsive pathways, like the hypoxia responsive pathways, get activated. That gives the signal to the cell, or cell, you are hypoxic, and now you need to increase all these processes, like angiogenesis, erythropoiesis, and so on and so forth. So we wanted to use that as our marker for hypoxia. So this is our study design, where we used a preclinical pig model of acute myocardial infarction. In a group A and B, we induced 210 minutes of LAD occlusion with or without 120 minutes of unloading. And we have harvested the heart and quantified the infarct size. In the group C and D, we did the same, but now with reperfusion. The idea being, all the studies in ischemia reperfusion injury is always done after reperfusion. We wanted to study what is the effect of LV unloading during ischemia and post-reperfusion. Then we also sampled the tissue from the infarct and non-infarct zones, sent it for LCMS, and did a metabolomic screen on them. So this is the infarct size quantification. So the first two bars are from the occlusion side, and the second two bars are from the reperfusion side, plus and minus unloading. We see after 210 minutes of LAD occlusion, we see hardly any infarct in there. And LV unloading was protecting the myocardium. We have a significant reduction in infarct size during ischemia alone. The major chunk of injury was only happening post-reperfusion. And by prepping the heart with LV unloading, we are significantly reducing the infarct size post-reperfusion. Coming to the oxygen sensor HIF, when we looked at HIF stabilization is a marker of ischemia. So during occlusion, when there is ischemic tissue, HIF was significantly decreased in LV unloading group, suggesting there is reduced ischemia with LV unloading. Post-reperfusion, we have an increased HIF1 alpha. Increased HIF1 alpha or stabilization of HIF1 alpha has been shown to increase angiogenesis and reduce post ischemia injury, and also increases myocardial recovery post-reperfusion. So we hypothesize, or we think this HIF stabilization post-reperfusion is beneficial for recovery of the heart. Now the second part is metabolic changes. So yeah, HIF is now reduced with transvalvular unloading, but is it also rescuing the metabolism in the myocardium? So when we looked at the marker as a marker of anaerobic glycolysis, lactate and glucose in these infarct zones, we found lactase was significantly down with LV unloading during ischemia, so as glucose. Also, other metabolites from this pathway was significantly reduced during ischemia, other than pyruvate, which might be from other gluconeogenesis pathway. We can create pyruvate. But other than that, the marker of glycolysis was significantly down. Now, the glycolysis was down, so is that the heart is compensating for the metabolites from some other sources? So we looked at the ATP and ADP generation in these infarct zones, and we found out the transvalvular unloading during ischemia is not only reducing glycolysis, but also shutting down, like unloading the heart and also reducing the workload on the heart by not producing ATP or ADP as a marker of increased metabolism. Then other markers, other targets of HIF and then association to glycolysis are PDK1 and GLUT1, which are target of HIF and also plays a role in anaerobic glycolysis. When we looked at the protein expression of these as confirmatory data to our metabolic profile, we found out PDK1 and GLUT1 was PDK1 was significantly down during ischemia, and post-reperfusion it's up in line with our HIF1 data. With this, I would like to conclude my talk. What we found, LV unloading reduces ischemic injury and attenuates reperfusion injury. LV unloading triggers global shift in myocardial metabolism during ischemia and favors reduced glycolysis. LV unloading reduces myocardial damage. In summary, what we showed here that LV unloading and prolonged ischemia is feasible, and by providing systemic hemodynamic support while normalizing myocardial metabolism and thereby making myocardial recovery more likely. With this, I would like to thank my colleagues at Tufts Medical Center, my mentor, Dr. Kapoor, and the funding agencies. Thank you for your attention.